All right, it's my pleasure to introduce our first speaker today, Chris Knupp. He is currently a third-year PhD student in the Aquatic Animal Health Lab at Michigan State University and is pursuing a degree in fisheries and wildlife with a specialization in fish and wildlife disease ecology and conservation medicine. He holds a bachelor's degree in zoology and a master's degree in fisheries and wildlife, both of which he obtained from Michigan State. His current research is focused on understanding the Flavobacterium psychrophyllum genetic diversity as it relates to bacterial cold water disease ecology and management. So Chris, I'll turn it over to you.
All right, great. Thank you, Dr. Phelps, and thanks to the organizers for giving us this opportunity to present our research. I'd like to first take a second and acknowledge my co-authors, Drs. Faisal, LaFrenz, Griffin, Soto, and Weens for their collaboration on the study that I'm presenting today, which is about these yellow pigmented rods or flavobacteria and how ultraviolet radiation may be able to reduce their numbers and therefore the risk of flavobacterial disease outbreaks and hatcheries in aquaculture facilities. So before getting into the details of this study, I want to take a moment to highlight an important trend in global fish production. This figure is showing you that we have historically produced fish for human consumption or for other products, mainly through capture fisheries, as you can see in orange. But as you can see in blue, raising fish through aquaculture has been growing tremendously over the past several decades and is now the primary way in which we produce fish for human consumption. So I constructed this figure based on data from the USDA, and it illustrates the impact of disease on US trout production. I know there's a bit going on here, but you can see we have millions of trout along the y-axis and years across the x-axis. What I would like you to focus on is the red line, which depicts the percent of fish lost to disease that had originally been intended for market. And so you can see that disease continues to be a significant impediment to the success of aquaculture. And according to the Food and Agriculture Organization of the United Nations, diseases caused by flavobacteria are among the top 5 contributors to these losses. So 2 of the most well-known and damaging flavobacteria are Flavobacterium psychrophyllum, which is one of the most significant pathogens of cold water fish such as salmon and trout, not only in the United States, but globally, and Flavobacterium columnare, which is believed to infect all freshwater fish species. With that being said, it's particularly devastating to the catfish industry, but also causes losses in salmonid aquaculture. And as you can see in these images, these flavobacteria are exceptionally damaging to their host to the point where they're causing significant gill and muscle necrosis, sometimes so severe that it's exposing the bone. Okay, so I just mentioned 2 flavobacterium species that we've known about and studying for a very long time, but it's not that simple because there are a multitude of novel and emerging flavobacterium species that cause disease in fish. In addition, there are also increasing reports of novel Chrysobacterium species, which is a genus in the same family as Flavobacterium, and they also cause disease in fish. Okay, so I just described the importance of these flavobacteria, some of which we've been studying for over 100 years. Unfortunately, we have very limited options when it comes to preventing and controlling flavobacteria. There are some vaccines, but their efficacy is variable, and so we continue to rely heavily on only 2 FDA approved antibiotics. Making things worse is that flavobacteria are mounting resistance to these antibiotics, and so we really need to develop non-antibiotic based prevention and control alternatives. So now I want to take a step back because if we want to develop alternative prevention and control strategies, we first need to understand how flavobacteria are entering hatcheries and aquaculture facilities. One important way flavobacteria enter facilities is through their source water, especially if that water is coming from a reservoir that contains fish infected with flavobacteria. So no matter how these flavobacteria manage to get into a facility, they can then take advantage of how many facilities are designed. And I know many of you are familiar with what I'm about to say, but many facilities have a flow-through serial reuse system where water flows from one raceway into another before leaving the facility. And this system is very important to consider from a flavobacterial disease transmission standpoint, because if flavobacteria are coming in with the source water, or if fish are infected with flavobacteria, either because of the source water or vertical transmission, then these flavobacteria can accumulate in the water and be passed down into the next set of tanks where they can cause additional flavobacterial disease outbreaks. So what can we do to prevent flavobacteria from entering facilities through source water, from being transmitted between raceways? One possible solution is by treating source water, facility water with ultraviolet light, which is proven effective for killing or inactivating other fish pathogens. Currently, however, we have very little understanding of what UV dose is required to kill or inactivate the wide diversity of flavobacteria that either cause disease in farm fish or inhabit their environment, which is obviously very important to know if we want to minimize the risk of flavobacterial disease outbreaks. In fact, there are only a few studies that have scratched at the surface of this topic, two of which only used one isolate from one flavobacterium species, Flavobacterium psychrophyllum, and together present apparently conflicting results, where the effective UV dose reported by Elmore was about 25 times less than what was reported by Hedrick. And a third study was performed in Hungary in the 1980s with Flavobacterium columnari, but the results of that study were inconclusive. And so we don't really know what dose of UV radiation is effective at reducing flavobacteria. And in this context, we know that there's a wide diversity of Flavobacterium psychrophyllum strains out there, as you can appreciate in this figure. And we're seeing more and more that some of these strains or complexes of strains appear to be linked to phenotypic characteristics such as host species preference and virulence. And so by testing the UV sensitivity of diverse psychrophyllum strains, we can better inform future management decisions aimed at reducing the risk of cold water disease outbreaks. So similarly, we know that there are at least 4 distinct genetic groups of Flavobacterium columnari that also differ in their phenotypic properties, but we don't know how sensitive they are to UV radiation, which again is important for developing appropriate disease mitigation strategies. So with this in mind, I formulated this overarching question so that we can understand if UV radiation can potentially be used as an effective tool for reducing flavobacteria common to hatcheries and aquaculture facilities. So my 3 hypotheses for this study were that all tested flavobacterial species would be reduced following UV irradiation, and overall the high UV dose would more significantly reduce flavobacteria than the low UV dose. Flavobacteria species would differ significantly in reduction at each UV dose. And my third hypothesis was that reduction would differ significantly within the species columnari and psychrophyllum. So to test these hypotheses, we selected 65 flavobacterial isolates. 43 of these isolates belong to 13 already described flavobacterial species, the majority of which are known fish pathogens. And as you can see, we chose multiple isolates of Flavobacterium columnari and psychrophyllum to capture the strain diversity I mentioned previously. In addition to these already described species, we also selected multiple potentially novel Chrysobacterium and Flavobacterium species. So the 23 chosen psychrophyllum isolates belong to 12 different strains, which I highlighted in red, and 3 of these strains, including 1078 and 275, are currently the most widespread in the United States, and together have been recovered from 17 out of the 23 different states psychrophyllum isolates have been genotyped from. These 3 strains also belong to the largest clonal complex worldwide, as you can see. We also chose strains from a variety of other clonal complexes that have different host preferences and therefore may also differ in other phenotypic characteristics, including sensitivity to UV. And we also included some strains that were not part of clonal complexes and may be a bit more unique. And so really the goal here was to try to capture a wide range of genetic diversity and incorporate some of the most widespread strains. We also incorporated 7 Flavobacterium columnari isolates that were genotyped by Dr. Ben LaFrance and are representative of the 4 different genetic groups. And so basically we were trying to do the same thing with columnary as we were with Saccharophilum, which was to incorporate a diversity of isolates from different genetic groups. So overall, the 65 flavobacterial isolates included in the study were recovered from many different sources. As you can see from the green portions of this chart, most isolates were recovered from infected fish, most of which were kept in hatcheries. The blue portions of this chart detail the number of isolates recovered from water, most of which you can see were also from hatcheries. The isolates were recovered from many different geographical locations. Most isolates used in this study were recovered in one of 11 different US states, but several isolates were recovered from Asia, Europe, and South America, as you can see in the table on the right. So to prepare the 65 flavobacteria for the UV experiment, each one was cultured in a liquid medium, incubated at an appropriate temperature with constant shaking for 48 hours, and then harvested using a centrifuge and diluted with sterile saline to a standardized optical density. So for the experiment, I used a collimating beam device, which is designed to test the sensitivity of a microorganism to UV radiation in a standardized way. The collimating beam uses several UVC LEDs that emit a consistent dose of ultraviolet radiation in the germicidal range. And you'll notice I set the collimating beam up on a shaking platform, and so that was used to keep the flavobacterial cells evenly distributed while the UV was on. And at the same time, and so using this device, so I tested I exposed the flavobacteria to either a low dose or a high dose of UV. And then, at the same time, I had a separate flavobacterial culture that was not exposed to UV, and this served as the control. And so then after UVing, I had performed serial dilutions, plate counts to quantify the number of surviving bacteria, and this experiment was performed in duplicate. So the effectiveness UV has on a microorganism is typically reported in two ways, either by log reduction or percent reduction, both of which are correlated, as you can see in this table. And so the results I'm presenting today will be in terms of log reduction, which is basically the log of how many flavobacteria were recovered from the control, which didn't receive any UV treatment, divided by how many flavobacteria were recovered after UV treatment. So for example, anytime you have a log reduction of 2, this means the number of bacteria were reduced 100-fold or by 99%. So now I'd like to get into some of the results of this study. So this box plot summarizes the log reduction of the 7 Flavobacterium columnari isolates following UV radiation at 25 millijoules, which is the black box, and 125 millijoules, which is the gray box. You'll also notice that part of the background of this figure is shaded in gray. This is the area where isolates were reduced by greater than or equal to 99%. Okay, so at the low dose, you can see log reduction range from 1.37 to 7.39, and on average, isolates were log reduced by 4.42, whereas at the high dose, log reduction range from 1.93 to 9.21, and on average, isolates were log reduced by 6.19. So overall, there was no significant difference in reduction between the high and low UV doses, which was not something that I had anticipated. And it's also interesting to note that even within this single Flavobacterium species, that there were significant differences in reduction among the tested isolates at each dose. So when we compare log reduction at the low dose for the different columnary genetic groups, we do start to appreciate some differences in reduction. So any groups with different letters are significantly different from one another. So for example, genetic group 3 was less sensitive to UV than genetic groups 1 and 2. And although not significant, genetic group 4 also seemed to be a little bit less sensitive to UV than genetic groups 1 and 2. So for the most part, this 25 millijoule dose did seem to work well and substantially reduced most of the columnary genetic groups. when we compare log reduction among the genetic groups again, but at the high dose, we see that for the most part, all groups are essentially reduced by greater than 99%, but that there is quite a bit of variation in response from the isolates in genetic groups 1 and genetic groups 4. And so genetic group may not be the best way to estimate the UV sensitivity of a columnary isolate, but it is an area that I think warrants additional study since we only examine 7 isolates. Okay, so now I'd like to go through a similar sequence of results, but for Flavobacterium or Psychrophyllum. So this figure summarizes the log reduction of the 23 Psychrophyllum isolates. So at the low dose, log reduction range from 0.17 to 8.18, and on average, isolates were log reduced by 2.29. Whereas at the high dose, log reduction range from 1.86 to 9.07. And on average, isolates were log reduced by 4.56. So unlike Flavobacterium columnare, Psychrophyllum isolates were more significantly reduced at the high UV dose when compared to the low UV dose. There were also significant differences in reduction among the isolates at each dose. So Flavobacterium Psychrophyllum does appear to be more resistant to UV radiation than Flavobacterium columnare, as the columnare isolates were on average reduced 100-fold more than the Psychrophyllum isolates at the same doses. And I'd also like to point out that at the low UV dose, only 1 Psychrophyllum isolate was reduced by 100%, whereas at the high UV dose, only 3 Psychrophyllum isolates were completely reduced. Now, when we group the Psychrophyllum isolates by strain type, which are given across the x-axis, you can see that there are quite a few strains, including all from clonal complex sequence type 10, which again is the most widespread and dominant clonal complex in the United States and world, that have isolates that are reduced by less than 99% at the lower UV dose. You can also see that at least one strain, sequence type 301, which is actually part of the second largest clonal complex in the world, is very resistant to UV radiation. In fact, one of the isolates belonging to this strain was only reduced by 37%. However, it is notable that there are other Psychrophyllum strains like these 4, which appear to be more sensitive to UV radiation. So this figure is structured like the last, but shows log reduction at the high UV dose. And what you can appreciate here is that at this 125 millijoule dose, nearly all strains were reduced by more than 99%, with the exception being sequence type 301, which, as you recall, was the strain that was very UV resistant at the lower dose. This figure combines the responses from both doses, so you can see them side by side. And what you'll notice is that most strains were more significantly reduced at the higher dose than the lower dose, as I indicated with the different symbols. And so with this experimental design and data set, it appears that when you increase the UV dose by a factor of 5, you reduce the number of Psychrophyllum cells by about 467-fold on average. Okay, so I just showed you log reduction for columnare and Psychrophyllum, but how about for all examined species and isolates, including columnare and Psychrophyllum? Well, at the low dose, log reduction range from 0.17 to 9, and on average, isolates were log reduced by 3.43, whereas at the high dose, log reduction range from 1.86 to 9.73, and on average, isolates were reduced by 5.34. And so overall, Flavobacteria were more significantly reduced at the high UV dose compared to the low UV dose, and within each of those doses were significant differences in reduction among the isolates. And so what's important to take away from this figure is that these 65 Flavobacteria that belong to more than 30 different species, most of which are coming from infected hatchery fish or hatchery water, were all reduced following UV irradiation, even at the lower dose. So I get to go theoretical for a moment. If I was a hatchery manager using a 25 millijoule UV dose on my water source, some Flavobacteria would continue to get through, but for others, they would be completely eliminated. And at 125 millijoules, I would find that nearly all flavobacteria would be reduced by a log of 2 or 99%. So this means that UV could be an effective tool for minimizing the risk of disease outbreaks caused by many different species of flavobacteria that are present in hatcheries and aquaculture facilities. So what we can conclude from this study is that UV radiation is a promising tool for substantially reducing a wide diversity of flavobacteria, and a higher dose will result in more significant reduction. With that being said, flavobacteria do vary dramatically in their sensitivity to UV radiation, including isolates of the same species. And so flavobacterial diversity is something we're probably going to need to consider when deciding on an effective UV dose. So now that we know that many species of flavobacteria common to hatcheries and aquaculture facilities are susceptible to UV radiation under laboratory conditions, the next logical step will be to test their sensitivity to UV radiation under more natural field conditions, meaning irradiating flavobacteria that are suspended in water from different sources that typically supply hatcheries and aquaculture facilities, such as rivers and streams, wells, and springs, all of which may have different levels of turbidity that could impact the effectiveness of UV radiation. And we also know that flavobacteria are excellent at forming biofilms. And so we also need future studies assessing whether flavobacteria coming from biofilms are more UV resistant. And so I'd like to acknowledge the USDA for funding this project, the Natural Resource Department in Michigan, Ohio, Pennsylvania, and Minnesota for supporting this research by allowing us to come and sample from their facilities, and my advisor, Dr. Thomas Locke. And I'll be happy to take any questions.
All right, thank you very much, Chris. Very interesting results, very good stuff. Feel free to drop your questions into the chat box, everybody, or you can unmute yourself and ask Chris directly. And I see your hand up first, Matt.
Hey, Chris, fantastic job as always. Really enjoyed that talk. Just two basic questions, and maybe I missed them. What do you know about like biofilm production in the different genetic groups of columnar? Has anybody looked at that? And then I guess my other question is, do you have any idea what the background levels of these flavos are in the source water that you'd be working with?
Yeah, those are good questions. I guess to answer the columnary one, I am not too familiar with how the different genetic groups form biofilms. I guess I know a little bit more for cycrophyllum. But I guess to answer your second question on the background of how much flavo is in the water normally, I can tell you, at least for cycrophyllum that I've looked into, there is not very many studies that have actually addressed how much you find under, either coming in with the source water or even, present in a raceway during an outbreak. I know there's at least one study I can think of that was done in Europe, and I think they found around 10 to the 1 in source water. And I think that was coming from like a river or a stream. And then they also had sampled some water, and they analyzed this using qPCR from a raceway during an outbreak. And they were finding about 10 to the 4 cycrophyllum cells. So I don't know, we know even less about other flavobacteria. So
it sounds highly variable. It's just kind of cool to me to think that if you're rolling at 99.5 to 99.9% reduction, and man, there's only 100 cells, per mil already in there or less, that you're doing a pretty good job. So I don't know, fantastic work, man. I really enjoyed that.
Thanks.
All right, Chris Whips, I see your hand up.
Hey, thanks, Nick. Nice work there, Chris. Seeing some of the differences between your strains with regard to resistance to UV got me thinking about, selection. Is there a concern that, sort of use of UV could actually select for resistance in the bacteria. I'm probably thinking more about a recirculating system where you'd have a constant potentially, these bacteria being exposed to UV. Maybe that's not as big of a deal with flow through. I'm curious what your thoughts are on that.
Yeah, that's a good point. And, I guess I could see it being potentially something that could happen, similar like antimicrobial resistance. I guess, and I say that because I have seen at least one study where they found that some flavos that were at like a higher elevation seemed to have, or no, it wasn't flavos, but it was a bacterial species, and basically at higher elevations where they would, be exposed to more of the UVC, that they did seem to be more resistant than, some of the ones at sea level. So, I think it could be something that evolution could drive towards. So, but, we don't know right now. So.
Sure. Cool. Yeah. Thank you.
All right. No questions in the chat. We'll go ahead and end there.